You should see nuclei all the way to the top, at least on some of the cells, if it's living all the way to the top. Okay, so that is stratified, and this one is, can you tell that it's stratified? I've got more than one layer of nuclei from the top to the bottom, and then this one is stratified. But so is this one, even though that's not its name. Multiple nuclei all the way from the top to the bottom. So it's a stratified tissue. Everybody okay with that? Okay, so now the first one that we're going to do is obviously going to be stratified and squamous. Squamous doesn't describe the shape of all of the cells. It describes the shape of the cells at the apical surface. Okay, so this is a stratified squamous epithelium. Before we even get into this, is this tissue going to be better at protection or transport? Yes, this is going to be protective, but it actually provides an impediment to transport, right? So typically speaking, this would be one extreme, whereas simple squamous epithelium would be the other extreme as far as protection and transport. Okay, so this tissue right here is going to be, hold on, I gotta get that down there. This is going to be one of the two stratified squamous epithelia. There are actually two of them. They are both going to be protective, but one of them has a source of moisture, so it doesn't need to be waterproof, and the other one does not have a source of moisture. Okay, so this tissue right here, this is a stratified squamous epithelium. Now, I want you to look at it and compare it and contrast it with its neighbor. What its neighbor is a keratinized stratified squamous epithelium. This one is called either moist or non-keratinized. Either word is fine. Contrast it with the next one. So what the next one is going to be is basically, it looks a lot like this, but then pile a whole bunch of dead cells on top, each of which made waterproofing protein keratin before it died. Okay, so what it's going to look like is this plus. So look at this one. Do you see that if I looked at it, it's a whole bunch of cells stacked on top of one another, and then a bunch of things that don't have nuclei on top of that? Yeah? So that's a keratinized stratified squamous epithelium. Does that make sense to you guys? Okay, so now let's look at a couple that are unfamiliar slides, but you should be able to tell which one they are. What is this one? Non-keratinized or moist. And how did you tell? Nuclei all the way up to the top, right? And now let's look at this one. What is this one? Keratinized. So I've got nuclei up to here, and then I've got a whole bunch of trash on top. Right? Does that make sense? Okay, so these two are similar to one another, but they are different, right? So let's talk about the body locations for each one of these and make sure that you guys actually understand it. So the body locations for this one is anywhere that you need protections, but you also have a source of moisture, okay? So places that have wear and tear, but also a source of moisture. So in this one, you are going to find places that have wear and tear, but don't have a really good source of moisture. So you tell me which one I'm going to find lining my oral cavity. Non-keratinized, right? Which one am I going to find on the epidermis? Keratinized. Okay, reproductive system. It's all about getting a little friction down there. What am I going to find lining the exterior surface of the penis? Keratinized. What am I going to find lining the vagina? Exactly, right? Protection, but a source of moisture, then you're going to have non-keratinized or moist protection, but no distinct source of moisture, you're going to have keratinized. Does that make sense? Okay, and there's the amount of keratin differs wherever you're looking at on your body. Lots and lots of abrasion, right? And also potential for needing grip, like on your hands and the soles of your feet, you're going to find a really thick layer of keratin. But in some areas where you have really, really thin skin and it dries out more readily, then it's going to look less like that and more like this, right? A kind of thinner layer. Go ahead.
So we learned in lab last week, so let me just clarify.
Sure.
When it's keratinized, does that mean it's cancerous? Is
that the only time it becomes keratin? No. Keratin and cancer don't have anything to do with one another. Keratin is basically just a protein that it made before it died, okay? So all epithelial tissue does have a higher predisposition to cancer than any other tissue. The keratin really isn't protective, but the melanin is. Is that what they were talking about?
Probably, yeah.
So we haven't done skin yet, but melanin is a protection mechanism for UV radiation. And so people with inherently darker skin, meaning I didn't have to tan to get that way, that's my inherent rate of melanin production, tend to have a lower rate of skin cancer than people with inherently lighter skin. Does that make sense? So although people of African descent, for instance, do get skin cancer, because cancer's a dick and anybody can get it, right? Do they get as much skin cancer as, for instance, I would?
No.
Or Stella, right? You better wear some sunscreen, friend. I hope you do.
We don't wear the sun shirt when we go out.
So that's your inherent rates, but we'll come back to that when we get to skin. Okay, so we've got those two, moist and keratinized, and now let's get to the other stratified tissues. So the other stratified tissues, this is a stratified cuboidal epithelium. It is stratified, but it's still not very thick. So you can still do both a little bit of transport. So do you think this is thick enough to be super duper protective? No, it's only 2 layers, so it's not that protective. You actually find this forming some glands. You actually find it sweat glands, mammary glands, salivary glands. A lot of times it'll have secretory capacity. So that's stratified cuboidal epithelium. And then stratified columnar, let me show you a couple other iterations of stratified cuboidal epithelium. Can you recognize that that's stratified cuboidal epithelium? Yeah, stratified cuboidal. How about this one?
Yes.
Yeah, that's stratified cuboidal. So don't be afraid of unfamiliar histology slides. As long as you get comfortable with looking at it and using the criteria in your brain, you're totally cool with these. Go ahead.
So the tissue around it, would that be considered?
This is all connective tissue. Yeah. I would imagine that this is probably dense irregular connective tissue. It's a little difficult to tell, but this one is a beautiful dense irregular connective tissue all the way around, right? That one is. Yeah? Yeah? So yeah, and so I could, for instance, use this and point to here. So on a histology quiz, which you'll have one posted on Friday right after the other quiz closes, I have to point to which tissue I'm talking about because, of course, there's more tissues on the slide. Right? Everybody good? Okay, so those are 2 are stratified cuboidal epithelium. And then one of the rarest tissues in the body that people still actually argue a little bit about is this guy right here, which is what? It's stratified, but it's columnar. Okay, so how can you tell it's columnar when you really can't see the boundaries of the cells? Oval nuclei, right? Taller than it is wide. Right, stratified columnar epithelium. This tissue is really rare in the human body. And by the way, every once in a while, you'll see it labeled pseudo-stratified. Your textbook uses stratified. I use stratified. Don't worry if you see it labeled something else. This tissue is stratified. So you really only find this tissue sort of dependably in the human body in the distal end of the male urethra. So let's talk a little bit about male and female urethras for just a second. So the urethra leads from urinary bladder to the external urethral orifice. In women, on average, it's about 1.5 inches long. Okay, varies, of course. Urinary bladder to the external urethral orifice. But in males, on average, the urethra is 8 inches long. I'm not telling you penises are 8 inches long on average. I would not lie to you in that way. Okay? I don't want you to have that expectation for your life. But from the bottom of the urinary bladder through the internal structures and then out the end of the penis is on average 8 inches long. And in males, it changes tissue type. So in both sexes, the majority of the urinary tract is lined with transitional epithelium. Okay? Ureters, urinary bladder, and the entire female urethra and the proximal end of the male urethra. So why does the males change tissue type? Well, because the male urethra actually has dual functionality, right? The male urethra actually has urinary tract functionality, but it also has reproductive tract functionality. And the reproductive tract functionality necessitates that there are different tissues that have different capacities. And one of the things that happens in the male urethra is that there are secretory capacities that actually cleanse the male urethra, change the pH of the male urethra so that the semen is exactly the right pH for fertilization, right? Does that make sense? And so this tissue type is related to that functionality. So only in the distal end of the male urethra do you find this stratified columnar epithelium. You don't really find it dependably at any place in the female. Good? okay? Even though it might kind of look like this. So that is lining the male urethra. Okay, and then the last of the stratified epithelial tissues is not called stratified, but it is stratified. It just sort of breaks the naming convention. And that is this one, which is called transitional epithelium. And this is the one that lines most of the rest of the urinary tract. Now, not inside the kidneys. That's a different kind of tissue. But ureters, and ureters lead from kidney to urinary bladder, which is your holding tank, and then urethra on the way out, all lined with this, except for that little portion that we talked about in the male, and this is called transitional epithelium. Now, can you tell it's an epithelium by looking at it? Yeah? Okay. Can you tell that it's stratified, even though it's not in the name? Yes? Okay. So why is this one not named properly? I don't know. Somebody didn't get the memo, but it's not. And also because the cells don't really look squamous or columnar or cuboidal. They're weird-looking cells. They're obviously stacked, though. So this tissue, its primary characteristic is that the cells on the apical surface look domed. Do you see that dome-shaped cells? Okay. And this is what it looks like when the urinary bladder is empty. But this tissue is capable of stretching. So the urinary bladder can hold a couple drops to a full pint of urine, right? So this is what it looks like on the lining of the urinary bladder when it's empty, okay? What do you think this tissue would look like when it was full? Yeah, it would look just like another tissue. Stratified squamous epithelium, a moist one. I don't ask it from you stretched because that's mean, okay? But it does look like a moist stratified squamous epithelium because everything kind of just gets stretched flat. So I ask you from an empty bladder, right? So we just kind of don't require you to know this one, what it looks like. So this is transitional epithelium, and it's found lining the majority of the urinary tract. Does that make sense? Okay, all right. And then the last one, which is PseudoStratified epithelium. The only PseudoStratified tissue that you are learning, and he just asked about this, is the one that is called ciliated PseudoStratified columnar epithelium. Although I don't think in your lab you're required to do all of those words. I actually got into the habit of doing all of those words because there's so much meaning in each one, right? PseudoStratified tells you that it's false stratified. I'll show you what that means in just a second. Ciliated tells you that it's got cilia all the time. Columnar is the shape of most of the cells. They're taller than they are wide, and of course it's an epithelium. You can write down whatever's in your lab book and whatever's in your textbook. It's fine. So why is it pseudo-stratified? What does pseudo mean? False. So what it looks like, and I agree, it looks like there are a whole bunch of cells stacked on top of one another. But when we actually look at it closer, we actually do the drawing instead, you will see that the cells are variable in size, but they are all attached to the basement membrane. So it's pseudo-stratified. Yes? That makes sense? You got some tall, skinny ones, and you got some short ones. So it's pseudo-stratified. And then the ones at the apical surface tend to have cilia on them, right? So pseudo-stratified ciliated columnar epithelium. Okay? So what do you know about this tissue by the fact that it has cilia on the apical surface? Yeah, it's going to move something along the surface. So we find this tissue, see if anybody can put structure and function together with for me. We find this tissue lining the upper portions of the respiratory tract. Anybody want to put that structure function together for me? What do you think it's doing up there? Yeah, it's moving mucus. And of course, we really need it here in Salt Lake City, especially in the winter, where we on occasion have the dirtiest air in the nation. What? No, we haven't really beat out parts of China yet, but we're working on it. Okay, we're Americans. We'll get there. Can't let China be first. And heaven forbid we actually vote for anything related to environmental legislation. Heaven forbid. Okay, I'm just waiting for all those damn old people to die the hell off so that you guys can be in charge because they jacked it up.
Yeah. You and me both, sister.
I am Gen X. I didn't do anything. I just make fun of both of them. Make fun of the boomers, make fun of the kids, make fun of everybody. So, but anyway, so what this does is this actually will wave up whenever you breathe in any respiratory irritants. They just continue to wave up, and then you get it caught in the mucus, and then you move it on up with the mucus elevator. mucus elevator. Do whatever the hell you do with it. Nobody wants to know what you do with it if you ever want to sleep with them. Don't let them know. It's true. That's like destroys marriages. Keep your mucus to your damn self. Okay, so that's pseudostratified epithelium. In addition, epithelium brings us into now the goblet cells. Okay, so goblet cells, they look like a goblet because this is what they actually are. So what you're actually seeing is a specialized cell, also attached to the basement membrane in this instance. And what it is it's going to stain relatively clear, and it's going to look skinny on the bottom because the nucleus is usually right there, so it ends up looking like a goblet. Does that make sense? Okay. And what it does is it produces these little vacuoles that don't usually stain with this staining technique. Those vacuoles have a protein in them called mucin, which when you mix it with water forms mucus. Okay. And you can put that in locations that you need mucus, right? So we know why we need it in the upper respiratory tract, right? Because it's actually catching things. But sometimes it's not about that. Sometimes it's about lubrication. Sometimes it's actually about secreting something that you need. So if you see goblet cells, kind of think about their functionality. So this is a different tissue that also has goblet cells. Do you see them? Right? What is the function of a goblet cell in this tissue? Any idea? The goblet cells. So first of all, figure out what tissue you're looking at, where you find it, and then the logic of the goblet cells there.
So simple columnar epithelium.
Good. Where you find it? Not this one. Nope. Yeah, lining the small intestine. Any idea what the goblet cells are doing? Yeah, but why? Why there? Lubrication, also digestive, breakdown, those kinds of things. That's what it's doing there. So when you see a goblet cell, it's going to produce a product and dump it onto the surface. And you can kind of start thinking about structure-function relationships when you do this. Okay, so now let's just make sure that you can figure out a couple of different things. Okay, so when you see cilia, you think structure-function relationship, but also you use it as an indicator. Because if you see distinct cilia, you don't have 12 tissues to choose from, you only have 2, correct? And what are the 2 tissues in which you run into cilia? Ciliated pseudostratified columnar epithelium and ciliated simple columnar epithelium. So really all you have to do in that instance is be able to tell this tissue, especially if I point at an area that looks distinctly simple, which I will do, because when they're under cell division, they look a little confusing, right? You have to be able to tell that tissue from that tissue. And does that tissue look simple any damn place?
No.
No. So what you need to do is you need to find a logic that will allow you to simplify the criteria every single time. And it gets really easy after you've practiced it a little bit. Okay, so those are your 2 ciliated tissues. And we ran into 2 tissues with goblet cells. Although FYI, you can't always see goblet cells in every single slide of any tissue. But if you run into really distinct goblet cells, it's probably which of 2 tissues? Simple columnar, not ciliated, right? And pseudostratified, right? Good? Okay. And then if you run into distinct microvilli, the only tissue that you found with distinct microvilli is what? Simple columnar, right? So you're using these criteria to help you check, to make sure that you have the tissue that you thought you had. Do you understand what I'm trying to say to you guys? So it's not just rote memorization. It's not looking at a picture. It's like, oh, I know that in this tissue I should see this, or I don't see this, right? To figure that out. Okay. Any questions? Go ahead.
Yeah, sorry for one for the apical surfaces for the
Yeah.
fill in the blank. It's ciliated pseudostratified
Columnar epithelium and ciliated simple columnar epithelium. Okay, so the last thing to talk about isn't something that you're going to identify on a histology slide, but it's something that's physiologically important, and that is the concept of these cell junctions. Okay, so you run into this in phys a lot, and we're going to run into it for the rest of the semester as well, but you don't see it on the slides. All right, so we looked at this figure last time, and so we identified a hemidesmosome last time, remember? That was kind of the spot weld that went from the epithelial tissue into like the basement membrane to like help it hold on. But what we didn't ever do is finish our metaphor of the hole tiling the floor. So we said, okay, So the subfloor is the superficial surface of the connective tissue that's deep to the epithelial tissue. And then we put down sticky stuff. The sticky stuff was what? Basement membrane, and the metaphor was, of course, the adhesive. And then we put down the individual cells, which were the tiles, right? But we didn't grout anything, so now we need to grout it, okay? And the grout is the way that the cells actually stay adhered to one another, which is, can we agree, kind of important on surfaces? Not that they have spaces between one another, but that you actually present an intact surface to the external environment, or even to the internal environment, right? So what we're going to have to do is we're going to have to stick the cells together laterally. So we call these lateral cell junctions, right? If I'm a cell, how do I hold on to my neighbors? By my lateral cell junctions, okay? So there's 3 different types of grout that you can use in this instance. Only 2 of them work with the metaphor. But if these guys right here in this front row are all cells that are in an epithelial tissue, and I am a pathogen, right? I have to have ways to keep me out here and you guys, which are maybe the blood vessels, right? Protected, right? So there's a couple of different ways that you can do that. Let's talk about the first one. The first one is this guy right here. The first one you are seeing right here. So this type of cell junction right here is called a tight junction. And what I need to do is you guys need to zip yourselves together with like a waterproof zipper, a solute-proof zipper, like a scuba zipper, right? Zip yourselves together. Would it be enough for them to just hold on to each other's arms? Or if I was a small solute, couldn't I go under or over, right? So this is called a tight junction, and this is what it looks like. Notice that it goes all the way through and holds on to the cell. A tight junction is about impermeability. It means that if I try to get between you, I can't. I would have to go through you, which gives you a lot more control, right? Okay. Now, it's not necessarily very strong because I could probably pull it apart from this end, but it is impermeable. Does that make sense? Okay. So a tight junction is about impermeability. The next one will be easy to understand because we already learned a hemidesmosome. Hemi means half a desmosome. This time, what I'm going to do is I don't care if I can like go under or over, but I don't want to red rover you guys apart, right? So what's going to happen, this is a desmosome. Desmosomes are not about impermeability, they are about strength. So what's going to happen is she's going to reach proteins and grab onto his cytoskeleton, and he's going to reach proteins and grab onto hers. They're going to have a really strong weld between them. Why is this a desmosome and this a hemidesmosome? Right, exactly. This is 2 cells grabbing, right? And this one's only got 1 grabbing, right? So this is a desmosome. It's kind of like the 2 are really holding on to one another, and it's all about strength. So if I yanked on you guys, I couldn't pull you apart, but I might be able to go under or over, so it's not necessarily impermeable, right? Okay. And then the last one, this guy right here, is really about communication. And so what happens with these are that the 2, so if I want these guys all to be able to communicate with one another, because this is going to be my mucus elevator for today. Sorry to do that to you, mucus elevator. And what I'm going to do is I want for them to be able to communicate laterally to one another. So you have a channel protein and you have a channel protein, and I'm going to stick them together, right? This will allow them instantaneous communication. So stick with me. Ready? If I am the nervous system and I tell her to wave because I want a mucus elevator, right? I tell her to wave. All of you guys are going to wave in sequence. You go first. There you go. Okay, so they're waving in sequence. And now I'm a respiratory irritant and you're waving me out. Does that make sense? Okay, so, and cells can use any combination of these. If they need strength and communication, they could use, for instance, desmosomes and gap junctions. Okay, and it's not just epithelial tissues, but it is really important for epithelial tissues. Other tissues can also use them. Are we okay? Go ahead.
Are gap junctions always going to be proximal to the desmosomes?
No. It can be any damn place that you want, and you don't even have to have desmosomes. They just stuck everything in this one picture. Okay, everybody good? Okay, so those are the lateral cell junctions. And then, so I want to give you guys a study tip for knowing when you're finished studying epithelial tissues, okay? So when have you mastered epithelial tissues? Well, I thought of all the kinds of questions I could think to ask about an epithelial tissue, or your lab instructor could think to ask about an epithelial tissue. And if you can do all of these with an epithelial tissue every single time that you see it, then you're probably finished with that tissue. So let's just look at any example tissue and think of the things that I could ask. So if you look at any tissue, do not miss the easy question of what general category of tissue this is. And there's only 4, right? Epithelial tissues, right? Connective tissues, nervous tissues, and muscular tissues, right? So a lot of times people won't expect that and they won't even answer it, right? So don't miss the big picture. So, and then the second question is what everybody expects, which is what is the specific tissue, right? And what is this one? Stratified squamous, be more specific. Moist or non-keratinized stratified squamous epithelium, right? Okay, so everybody expects that question, and a lot of students never go further than that. And that's part of the reason that they hate histology, because they never actually figured out what they could be asked. But there are a lot more questions I can ask you out of this. I could also ask you, let's say, any structure. What is this? The lumen. What is, even though this isn't a great question to ask, what is this boundary right here between the epithelial and the connective tissue? So those are fair questions. And then some tissues have more things for me to ask you, like, where are you? Like, what is this structure? What is this structure? Goblet cell. So those are all fair questions. Good. Go ahead.
Sorry, is a goblet cell lumen as well?
No, it's not. It just doesn't stain. It's actually a full cell. It's not empty, right? It just doesn't stain with that protein staining technique.
Do you see where I get confused
Yeah,
with that?
Yeah, you just get used to looking at it in context. It's within the epithelial tissue, so it's a goblet cell and not a lumen. But I can see totally why you would think that. It's just not empty. What it is, let me show you. Maybe this will like reinforce it for you. Okay, what you're actually looking at in a goblet cell is this. Remember that nucleus is right there? It's not an empty cell. It's completely full. It's just full of bubbles that don't stain with that staining technique, right? So that's what you're actually looking at with a goblet cell. Okay, and then don't forget that I can also have you name the surfaces. So if we go back to this tissue for just a second, there's 2 different questions that I could ask pointing to this. Listen to the different phrasing so that you get the question right. If I say name this structure, what's the answer? Cilia. What if I say name this surface? Apical surface. Yes, that makes sense? Okay. And likewise, there's 2 different questions that I could ask here. Name this surface. Basal surface. Name this structure. Basement membrane. Exactly. So it is my job to write a clear question, but you have to answer the question that I asked, or else it's wrong, and I'm not going to argue about it. Okay? So you have to read it, and I'm not trying to be tricky. I just there are words that mean things, right? And I'm not responsible for your assumption. Go ahead.
So where's the apical surface on this?
Apical surface is right here on this one. It's right here on this one. It's the one that's always sort of the boundary of the free surface or the lumen. Yeah? And it can be upside down. The apex doesn't have to be at the top. It just has to be near the free surface. Is that okay? Okay. And then this one is what students are bad at without practicing. And I want you guys to try to get better at it. And I want you to see what we're looking for first. First off, you are going to need to, for instance, name a body location. But it has to be specific, because what you are doing when you name a body location is playing Magic School Bus, trying to get me there, okay? So if I say name a body location for this one, and you say small intestine, you didn't Magic School Bus me to the right tissue, because can we actually agree that the small intestine is made of more than one tissue? So what do you need to say to be specific? Lining the small intestine. Exactly, lining the small intestine. Small intestine will, of course, have, just see if this makes sense to you, epithelial tissue lining it. Then it's going to have a connective tissue. Then it's going to have some smooth muscle. Then it's going to have another connective tissue, right? So you have to actually say lining when you're talking about epithelial tissues most of the time. Now, with this one right here, name a body location for this tissue. Yeah, epidermis, the whole damn thing is made of this tissue. So that's Magic School Bus enough, right? But what about this one? lining the oral cavity, vagina, a whole bunch of different places, right? So you just have to be specific enough that I know that you know where it is, right? And that means that you didn't memorize it. That means that you actually understand it. Is that okay? Okay. Now, why the hell do I do this for you or to you when most students don't do well on this? One of the only things that you will retain of histology is actually the body location. You don't actually remember the name of necessarily all of the damn fibers, but you do, for instance, remember later on, oh, okay, well, the vagina is lined with this thick tissue, right? That's what it's lined, that has a source of moisture. So I'm going to make these quite often on your first test. They're fill in the blank questions. So I say the tissue, I ask you a couple of things, and I ask you a specific body location of the tissue. Because I don't know which one you're going to memorize, do I? I don't know specifically that you're going to memorize lining of the vagina or lining of the oral cavity, and they're both right. So it's going to be a fill-in-the-blank question. And some of you guys, usually on average of 25%, will leave all of the body location ones blank, which is silliness. You can remember where it's found. If you don't remember where it's found, it's arbitrary that you memorize the tissue at all. Go ahead.
Is lining of the mouth also correct?
Sure, yeah. Oral cavity, you know, you can get fancy if you want. As long as, I mean, the school bus got to the right place, right? Didn't it? Okay, that's what I'm concerned about right now. Okay, everybody good? Okay, and then the last little tidbit is don't forget that you are dealing with cells, right? It's not like completely unfair for somebody to ask you what that is. That's a nucleus, that's a cell membrane, that's some cytoplasm, right? So don't forget you're dealing with cells. And if you can do that, again, over multiple iterations of the same tissue, then you're kind of done with that epithelial tissue. And then you can move on. And I'll do the same thing for you with connective tissue, which is actually a little harder. There's a lot more to remember on connective tissues. Okay, and last tidbit before we move on to connective tissues is what about the histology slides that are epithelial tissues, but I don't make you memorize the slides because they're hard. So, but we do need to kind of acknowledge, just for anatomical education, that glandular tissue is made of epithelial tissue, even if you don't have to memorize what it looks like. And let's figure out what the hell we're talking about. Glands all over your body are made with epithelial tissue. It ends up being clinically important because, of course, that predisposes them to cancers because of high rate of cell division. It also means they're pretty good at repairing, right? So what is a gland, first of all? A gland is any cell. It can be unicellular, like actually a goblet cell is considered a gland, to multicellular, that produce and secrete a product. Good? That makes it a gland. So he's louder than I am. So there are two big categories of glands, and the two big categories of glands have to do with where their product ends up. I'm going to give you sort of where their product ends up, and you match the names. Okay. If you secrete a product onto a surface, you have a different name than if you secrete a product into the bloodstream. Which one of those is going to be considered an exocrine gland? Onto a surface, yeah? It doesn't have to be external surface, but it's going onto a surface, right? And the other one is going to secrete its product into the bloodstream, and that's going to be the what? Endocrine gland. Okay, so let's take just a minute and see if you guys can brainstorm some things that would be considered exocrine glands, even if you didn't know that before. Exocrine glands.
Salivary.
Salivary glands, yeah. Sebaceous glands. What?
Merocrine.
Yeah, merocrine, which is part of your sweat glands, right? Okay. That's pseudoriferous glands. What else? How about lacrimal glands? What are those?
Tears.
And what are they? Endocrine or exocrine?
Exocrine.
Exocrine, exactly. What about all of the goblet cells that you've met? That would be exocrine. What about mammary glands? Exocrine. Exocrine glands. Yeah, exocrine glands. That makes sense to you guys? Okay. Mammary glands are actually just highly modified sweat glands. And that would sell way less porn, don't you think? Highly modified sweat gland magazine. So, not that anybody buys magazines anymore. What the hell am I talking about? Okay. And then endocrine glands secrete a product into the bloodstream. The product is, hint, generally called a hormone. So tell me some endocrine glands. Gland. What secretes it? Not what is secreted. Thyroid was good. Pituitary gland, good. Adrenal glands. What secretes insulin? She said insulin.
Pancreas.
Pancreas. What secretes testosterone? Testes, right? Good. That makes sense? So those are endocrine glands. Does anybody know, I'm not going to test you over this, but maybe you know. Anybody know one gland that is actually both endocrine and exocrine? It secretes digestive enzymes into the digestive tract, which makes it exocrine. It secretes hormones into the bloodstream, which makes it endocrine. Huh? Mm-mm. Not that one. It's pancreas. Because the pancreas secretes digestive enzymes, proteases, lipases, but it also makes insulin, glucagon, and some other hormones. So it's actually both. It's one of the few exceptions to the rule. Does that make sense? Okay. So what do endocrine glands look like? You don't have to learn them. They look like blobs of cells with a whole bunch of capillaries in them. They're really hard for anybody who's not a really good histologist to actually identify. But let's look at what exocrine gland looks like. An exocrine gland is going to lead to a surface. So see if it makes sense to you. Where are you little bastards? Oh, they're at the bottom. See if it makes sense to you that exocrine glands, now, you do not have to know all of these individually. You just have to be able to recognize them as exocrine. And so what you are seeing is they are going to lead to a surface. So what you tend to see is they are curved below the surface and have a duct that leads toward the surface. From a top view, the duct would look like a pore. Yeah? Does that make sense? And if you looked at these up close, can you not kind of tell that they're epithelial tissue anyway? Don't they look like highly curved epithelial tissue? Right? Without having to know their individual names of the different types, right? So they secrete their products onto a surface through a duct that leads to the surface, right? That makes them exocrine. Is that okay? Okay. So for me, you don't have to know the individual names of any of these. You just have to be able to recognize that they are an actually exocrine gland. Okay. Good. All right. So now we are ready to move to connective tissues. Okay. All right. So connective tissues are generally harder for students than epithelial tissues. And there's a reason for that. First of all, nobody decided on a consistent naming scheme that they ever use. They're just called whatever the hell they're called, right? And that's not helpful. Second, they're super duper variable in both structure and in function, okay? So that is confusing. So first, let me explain to you why the hell blood, bone, fat, ligaments, tendons, cartilage are all considered connective tissue. Because as an adult, they don't have a whole hell of a lot in common. So the reason that they're considered all the same kind of tissue is because they're all derived, and you don't care that much, but they're all derived from this tissue that develops relatively early during embryonic development called mesenchyme. And then mesenchyme makes all of them. So if you can track it back to mesenchyme, it is a connective tissue. Also, how do you tell that it's connective tissue? Well, even though they don't really all of them have a lot of things in common. They are obviously not epithelial tissues. They don't have surfaces, really, right? Muscular tissue is really easy to identify, so they're not that either, and they're not nervous tissue. So anything that doesn't fit into those categories goes into this big bucket that goes connective tissue. So, and we're going to look at this in a little bit more detail in just a second. So, they are the most abundant, the most common tissues in the human or mammalian body. They're also the most diverse. They're super duper widespread, although on a good day, should you have connective tissue exposed to the outside of the body? No. It should always be covered by epithelial tissue on a good day, right? So they are usually really, really vascular. What do I mean by that? Lots and lots of blood flow. There are exceptions. Cartilage is avascular in the middle, and that matters because how well does cartilage heal? Anybody get this part of their ear pierced at one point? Is there still a hole under there? Oh yeah, there is. There's a hole under there. There still is a hole under there. So because cartilage hardly heals at all because it's an avascular connective tissue. Anybody damaged cartilage on accident? Is it still damaged? Still cause, yeah, it's going to cause problems for your whole damn life. It doesn't heal all the way almost ever. Ligaments and tendons are also, they're not avascular, but they're really poorly vascularized, so those don't heal very well. All of the other connective tissues tend to be really vascular. Those two are kind of the exception to the rule. And then the thing that's a huge characteristic of the vast majority of connective tissues is that you are going to have cell, a bunch of stuff I need to name, and another cell. Unlike epithelial tissues that cell, right next to one another, right? Widely scattered cells with a whole bunch of stuff that needs to be named between them, right? So that's true of these, right? But is it also true of this one, even though this is a really different tissue? Cell, a bunch of stuff that I have to name before I get to the other cell, right? So that's one of the characteristics that's usually true. All right, so let's talk about the categories of connective tissue and then a little bit more about functionality, and then we'll move into the specific ones. So the categories of connective tissue go kind of back to here. So you don't usually have to categorize them very much in lab. Most of the time they don't make you do this, but I want you to understand like how they have things in common and things different because it helps you remember them. So basically what they have in common embryonically is that they're all derived from mesenchyme. Mesenchyme is a tissue that's just like embryonic connective tissue. I do not hold you responsible for this slide, but I'll just show you. This is the connective tissue that gives rise to most, if not all. There's also another embryonic one called mucous connective tissue. It doesn't look like a damn thing, right? I mean, it's definitely not an epithelial tissue, right? But other than that, I don't see really anything. I can't even, like, they say those are immature protein fibers. I don't even see anything there. I don't know what the hell you're talking about. But this weird-looking sort of non-specific tissue will give rise to all three categories of adult connective tissues. And basically, when you're dealing with adult connective tissues, what you have is you have solid connective tissues, bone, you have liquid connective tissues, blood, and then you have all the squishy ones in between, right? Does that make sense? And those are these that you're seeing right here. So the solid ones are going to be supporting connective tissues, and that will include bone and cartilage, right? Solid, yeah, tissues. And then we're going to have the fluid connective tissues, which is pretty easy because that is blood. There's only one of them. All of your other body fluids, think about this. Why is blood a tissue and urine isn't? Why?
The blood has cells and
Exactly, right? So, right, I mean, it doesn't seem initially intuitive that it's actually a tissue, but it's cells with stuff around it and it's functioning together, so it actually is a tissue, right? And your urine doesn't really have any appreciable number of cells in it unless you have an UTI, right? You pull a few epithelial cells out on the way out, but other than that, not really much going there. So fluid connective tissue, which is just blood. And then we have the hard ones. The hard ones are the squishy connective tissues. And these are called connective tissue proper. And they have less water than blood and more water than bone, right? So that's how those are, okay? And with the connective tissues proper, you have two subcategories, and we'll get to them later, loose versus dense, depending on how many fibers are actually packing the matrix. Okay, so what are the functions of connective tissue? Well, look at all of this crap. Any single thing that blood does is a connective tissue function. That bone or cartilage does, or that fat does, or that ligaments and tendons do. So the functionality of connective tissue is just as varied as the structure. So is this a complete list of connective tissue functions? No, it's just to give you an appreciation for how many different things it could do. Okay, so now we are going to get into the basic organization of a connective tissue. So when you look at any connective tissue, what you are going to see, and there are of course exceptions because there always are, is you are going to see cells, fibers, and then the stuff that is neither cell nor fiber, which is called ground substance. Okay, so you've got cells. You're going to need to label the cell in every single connective tissue. You have to practice that to get good at it. I'll help you with it. Okay, you're going to see fibers. The orientation of the fiber and whether the fibers are bundled or not and what kind of fibers will tell you something about the structural stability of the connective tissue. And then you're going to see this stuff, which is neither cells nor fibers. And ground substance is such a general term that it doesn't mean a damn thing. It means I'm not cell, I'm not fiber, what am I? Well, if this was blood, and it's not, that would be 90% water. But if this was bone, that would be mostly calcium crystals, right? So the ground substance is the stuff that is neither cells nor fibers, and it really varies a lot from tissue to tissue, what's actually in there. Is that okay? So let's look at individually. So what do you find in every connective tissue? Well, you find cells, and the cells are generally going to have one of 3 different suffixes, okay? And so you may find blast cells, site cells, or clast cells, okay? If you find a blast cell, it's telling you something about functionality. A blast means what? Bud or precursor, exactly. So if I find an osteoblast cell, it's probably going to make some bone, yeah? If I find a fibroblast cell, what do you think it's going to make? Fibers, exactly, of some sort, right? So those are blast cells. Blast cells are usually really active in making stuff. That's what those are doing, okay? So in bone, you have osteoblasts. In dense regular connective tissue, did you guys learn this yet? It usually doesn't stick the first time you hear it. These cells are called, right here, fibroblasts. What have they been doing their whole damn life? Making some fibers exactly around them. Good, okay? And then sometimes when a cell is more mature and it can chill a little, it's called a site cell. A little less active in making stuff. So in bone, you would call it a what? Osteocyte. What would you call it in cartilage? Chondrocyte. In fat? Adipocyte. Exactly. Good, right? Okay. So sometimes they're called site cells. And then although in this set of notes you don't run into them, just because they are not in a lot of the common histology slides, they are a thing. You can often run into clast cells. And clast cells, clast means to destroy or break down, because sometimes to change the structure of a tissue, you need to do destruction before construction, right? So like, for instance, you'll run into osteoclasts when we do bone, right? So if you started working out, you would need more bone tissue at certain points than you need at other points, right? And so you would break it down in some places and build it up in the other places, break it down with osteoclasts, build it up with osteoblasts, right? Okay, so, and then you're going to run into matrix. And the matrix is simply the area that is not the cells. So if this is a cell, this is a cell, this is a cell, everything that's not cells is collectively matrix, but the matrix is composed of fibers and ground substance, okay? So go ahead.
What was the fill in the blank?
I have no idea.
Those fibroblasts.
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 Blast.
Yeah. So, ground substance. So again, just ground substance for a second. What is in the ground substance? Well, it totally depends on the tissues. Usually it's got some kind of crystallized ions or some sugars or maybe some proteins that are not fibrous proteins in there. So it's got something in there because it's not empty, because you don't really have empty blank spaces. And also some degree of water, right? So the ground substance is water plus something that was made by the cells or put in there. And then let's talk about the protein fibers. So the protein fibers are all of these long skinny things. And of course, they were probably made by the cells around them. And so you really have 3 categories of protein fibers, and they have 3 different sort of strengths and benefits associated with them. So when you look at a connective tissue, what fibers are in there and whether they're bundled or individual will tell you something about the structural integrity of the tissue. So let's look at the 3 types of fibers. Collagen fibers, a good metaphor for those would be like the fibers that you use to make a rope. Individually, not super strong, but collectively, if you get thousands of them together, they're really strong, right? Are they stretchy though? No, they're not stretchy, right? So they're strong, not stretchy. And if you run them all parallel to one another, you'll only get strength in this direction. But couldn't you use the same fibers to make a net and get strength in a bunch of different directions, right? So that's collagen fibers. Typically speaking, collagen fibers are long, thicker than these, right? And they don't branch. Usually, but not always, they stain pink. Don't ever use color as your only indicator because there's a bunch of different staining techniques. Okay? And then the next ones are these. They're thinner, They stain darker, and quite often they will branch. Okay, those are called elastic fibers. Elastic fiber is a good metaphor for those. It's the like rubber band elastics that you use with your braces, right? Individually, not super strong. But what if you had 1,000 of them? They would be really strong collectively, but they would still have some snap to them. So instead of a rope, sometimes you need a bungee cord. Yeah? So you can use them to actually make a bungee cord. Does that make sense? Okay. So they've got some stretch. And then the last one, these don't stain very well because they're so thin. You can only see them if you specifically stain for them. These little guys, they're called reticular fibers. And reticular fibers are, a good metaphor for them would be like a hairnet. So they're not really very strong, but what they can do is they can hold the general shape of a squishy thing. Like, why does your liver kind of keep liver shape even though it doesn't have anything like holding it on the outside? It's because running all the way through the middle, it's got sort of this hairnet holding onto its general structural shape. Does that make sense? Okay. Now, although these are present in more than one tissue, you only end up labeling them in a single tissue. Anybody know what that tissue is? It's called reticular connective tissue. It's the only one that you can super duper see them in because they stain it specifically for that. Is that okay? Okay. So, now we will get into the types of connective tissue. So we already talked about mesenchyme. Mucous connective tissue is similar to mesenchyme. I don't hold you responsible for either of those slides. Okay, not identifying them visually. You do need to know what it has to do with the story, though. Okay, so when we get into adult connective tissues, the adult connective tissues are connective tissue proper, supporting, and fluid. We're going to do the hard one first because you guys need a little help with this one. And connective tissue proper has a further subdivision, and the further subdivision is loose versus dense. And to understand what I mean by loose versus dense, I want you to look at this one versus, watch, this one versus this one, and tell me what is this one, dense or loose? Dense. And then the other one is loose, yeah? And so what you're talking about with loose versus dense is like how much of the matrix is like packed with fibers, right? That tells you kind of loose versus dense. Does that make sense? Okay. So the loose connective tissues, do you think that those tend to be a little stronger or a little weaker than the dense connective tissues? A little weaker. They have fewer fibers in them, right? Okay. So that's sort of the difference between the loose and the dense connective tissues. So let me ask you on this slide, okay, what is this tissue, loose or dense? It's actually dense. It's dense because I have big old chunks. I'm not saying I can't have any spaces, but most of the space is actually filled with this like fibers that you can see really packing the space. So let's do a couple more, see if you can figure out whether this one is loose versus dense. What about this one? That one's dense. Yes, that one's dense. That one's a dense connective tissue. Good. You'll get better at it as we practice a little bit. Okay, so the loose connective tissues usually have more space, more ground substance, a lot more. You don't tend to see the fibers bundled as much, like not as much bundled fibers. And so they're really, really common, not super duper strong comparatively. They're usually really vascular. They've got lots and lots of space for blood vessels to be running through them. And they're kind of used for packing under and between things. And sort of your classic loose connective tissue is this one right here, which is called areolar loose connective tissue or loose areolar connective tissue. Areola means opening or space. So this has got lots of openings or spaces within it. Okay, so areolar loose connective tissue. And this is a tissue that if I learned any connective tissue, I would learn this one because it's so easy to write questions off of it because look, you can really easily see collagen fibers, elastic fibers, right? You can see cells, you can see matrix. I could ask you whether it was proper, supporting, or fluid. I could ask you whether it was loose versus dense. So let's make sure you guys feel really comfortable with this one, okay? Loose areolar connective tissue, or areolar loose connective tissue. So this one you have run into before if you've ever skinned anything. Have you ever skinned a fish or a chicken or a rabbit or a human. I've skinned lots of humans now, more than animals at this point. That's very weird to say. But yes, I have. Have you ever skinned anything? You haven't? What the hell? You need some redneck experience like me, right? I skinned some squirrels in my day. Don't think I have not. Okay, so
That's your Texas roots talking.
it is my Texas roots. It's very important to keep them. I tried to pretend that I was not a redneck before, but I figured out that leaves a hole, a gaping hole in the population. This is my role. So when you skin something, what you will see is go home and skin a damn chicken. They don't come with the skin off of them. So when you skin something, even if you skin a fish, what you will see is there's a natural sort of separation between the skin and the deeper musculature, right? And you can feel it on yourself right now by doing this, right? Pull on an area that doesn't have really any subcutaneous fat to speak of. And feel how it's got a little wiggle to it, but it's also got a little snap back in it. Do you feel that? Okay. That is lucerarelar connective tissue. Now, when you skin something, it won't be yourself, but when you skin something, what you see is you can pull up on the skin and it will separate from the muscle, but you can see the fibers popping. Little silvery fibers popping. What you're doing is pulling apart this tissue. You're pulling it apart because it is blank to the skin and blank to the muscle. Deep to the skin and superficial to the muscle. Yes? Okay. So this tissue is lucerarelar connective tissue. It's kind of used as sort of a general packing between things material. So it is soft and it is pliable. It's got collagen fibers for a little strength, but not bundled, right? Elastic fibers for a little snapback, but not dramatic, right? Elastic fibers. It's also got reticular fibers to sort of just hold a general shape in there. You can't usually see the reticular fibers in a histology slide just because they don't stain well, but it doesn't mean they're not there. They are there, okay? So I wouldn't make you label them. And then it has boatloads of cells, but it's such a weird tissue that it's got all kinds of cells in it. You can label them fibroblast if you want to. That's fine. It's kind of a fib because you really don't know what the hell they are because you can't quite tell yet. But there's definitely some fibers in there, so fibroblast is kind of safe. If you can't remember what a cell is called in a connective tissue, guess fibroblast, right? Because, you know, it might be right. Could be, okay? So you can guess fibroblast cells. I learned them as like, okay, until you can tell, you just call them interstitial cells, which is cells that's in that fluid, interstitial fluid. But in addition, since this tissue is like deep to like your epidermis, it's also deep to all your moist membranes, like lining your entire digestive tract and your urogenital tract. And there's room in here. Does it surprise anybody that quite often white blood cells move in there, take up residence, and they're like sentries? Because isn't this a super common place to get exposed to pathogens? And you got room? White blood cells will move in and sort of take up residence, just in case you get exposed to something. Isn't just deep to any epithelial tissue that's exposed to the outside of the body a fantastic place to put a white blood cell? Right? It is. It's a fantastic place. So you're going to run into those sometimes. Every once in a while you get adipose in there, and we'll talk about those later. So this is the glue of the human body. You find it like under like most epithelial tissues in the human body. So where is it? It's under your external epithelial tissue. Smack myself in the face. External epithelial tissues. It's under the lumen of your blood vessels. It's under epithelial tissues lining your lungs, bladder, muscles, lymphatic, just damn near everywhere. So this tissue is super duper common in the human body, and you need to be able to recognize it. So this is lucerarelar connective tissue. I'll show you a couple more pictures of it just to make sure you got it. Okay, a couple more pictures. Can you recognize it even if it's a different level of microscopy? Can you still recognize that it's lucerarelar connective tissue? How about this one? Right? It's pretty easy to recognize. Go ahead.
Is this what, I'm just trying to remember from like high school anatomy, is this
Yeah.
what like fascia is made of?
Mm-hmm. Mm-hmm. Very good. Yes, it's what fascia is made of. Usually. There's a couple of different kinds of fascia, but the most common fascia is made of this one. Everybody okay with that? Okay. So make sure that you can recognize this one, because if I run out of a quiz question or a test question, I'm going to write one about this because it's so damn easy for me. There's like 72 easy things I could label on there, right? And so I get tired sometimes, and sometimes I'm having a cocktail and I want an easy question because I need to write a new one, right? Okay, so Now, the last thing I'm going to point out to you guys today is something that seems really counterintuitive, but it totally makes sense if you really, really think about it, which is that adipose tissue, this weird looking tissue that kind of breaks the rules that you just learned about connective tissue, really, God dang it, where'd it go? Where really all adipose tissue is this tissue, and then start getting fat. Okay, so this tissue, this is a stylized diagram of the same tissue. And so in this subcutaneous layer, we all know that you can have more or less adipose that moves into it, adipocytes that move into it. So what happens is this, although it's not staining on this figure, this has a few adipocytes in it, especially if it's subcutaneous right here in the subcutaneous layer, right? Okay, so what happens is it can end up looking like this, really kind of looking like this one right here, which doesn't look at all alike. So let me show you how that actually occurs. How that actually occurs is adipose tissue is really loose areolar connective tissue plus a whole bunch of cell division in your adipocytes. So it starts out looking like loose areolar connective tissue. It's got fibers, it's got some other cells right here, right? Like different kinds of fibers, right? And then it is, you had puberty, right? You hit puberty and you didn't used to have any hips, but then you get some, right? Okay, so you hit puberty and you had a few of these big adipocytes. Adipocytes are mostly non-staining simply because this is lipid and I use a protein staining technique and it doesn't stain this lipid. Okay, but what happens is when you start gaining weight, these will start to undergo huge, huge cell division. And when they do, they will push all of these other cells between them and fibers between them. So most of what you end up looking at is something that looks kind of like chicken wire, right? But what it really is loose areolar connective tissue plus boatloads of cell division in your adipocytes. That's why it's a loose connective tissue. Because it started out like this and ended up like this. It's still got all of these fibers, even though, can you label the collagen fibers? No, because they're smashed between the adipocytes. Can you label the elastic fibers? No, you can't. Are there fibroblasts in there? Yeah, but they're smashed between things and you just can't see them very well. So when you actually look at it really up close, it's actually this tissue that's sort of cell. Does that make sense to you guys? That's why it's categorized as a loose connective tissue. So we have areolar, and we have adipose. And let's just learn a little bit more about adipose and then I'll let you guys go. So,
If we were given the slide, we'd label it like adipose.
Yes.
Loose connective.
Just adipose, and then if I asked you which category it fit into, loose or dense,
Okay.
you would tell me loose or dense.
Okay.
But it's just actually called adipose tissue. So do you kind of understand how adipose got to look like it did? Okay, so what is adipose's job? It's insulation, it's cushioning, it's a major energy reserve. It's really a specialized tissue for energy storage. It has very little visible matrix because the adipocytes take up most of the space, right? So even though it looks like this is all matrix, it's not, because what am I actually pointing to here? Is this a lumen? Is this matrix? What is it? It's an adipocyte. It's actually what's called a lipid vacuole inside an adipocyte. This portion right here, it's called a lipid vacuole. And that is not empty. That is full. That is full of fatty acids that I could burn later if I needed them. Does that make sense? So it's got that lipid vacuole inside of it. And a single cell is called a what? Yeah, so what is this guy called right here? Adipocyte. An adipocyte. And the adipocyte has what we call a signet ring shape, meaning if you look at a high school ring from the side, the stone is on one side and it's skinny everywhere else. So usually it looks really skinny with one lump for the nucleus on one side. So it's called a signet ring shape. And one of the reasons that adipose is kind of a jerk is that when it lays down under your skin, it doesn't lay down nice and smooth. It lays down lumpy and little fat blobs called lobules. And if your skin is relatively thin, it will follow and you get cellulite, right? And it has to do with not necessarily how heavy you are. Of course, the more adipose you have, the more likely you are to get cellulite, right? But do you know any skinny people with cellulite?
Yes.
Yes, you do. You might not know that you know them, but you do know them. It has to do with how thick your skin is, whether you actually see it, and also how much adipose you have. Okay, last little tidbit before you guys leave. The tragic thing about adipose tissue is that your body can always be motivated to go through adipocyte cell division. You can always, always, always make more of them. It will always make more storage facilities for you. But once you get your storage facilities, do you ever get rid of them? No. All you do is burn the lipids in the lipid vacuole and keep the cell so that the next time you gain weight, it's easier to store it. So the first time you gain weight, it's usually a process, right? Then you lose it, and then the next time you gain weight, it's like instant, right? You ate like one extra bag of Doritos over the course of a week, and you gain 17 pounds. No, I'm just kidding. That's not how that happened. Okay, I will see you guys on Tuesday. Don't forget your quizzes. Don't email me if you do forget your quizzes. Okay?
Is the other one graded?
No, because I forgot and left it open for way too long, and I can't close it now because people expect it to be open. So it was just a mistake. Just do it, and I'm not going to close it until it says it's going to close.
Okay, yeah. Wait, not the syllabus one. This is an additional one.
The other one is due tomorrow. The anatomical terms one is due tomorrow. I will post a histology quiz for you, but it won't be due until after I've finished histology. Okay?
Okay, which two do I need to do?
The two that are up there.